Thank you, Dr. Conley, for inviting me to speak. You lead the Cancer Diagnosis Program, and now for something that is really rather different, we have had three wonderful speakers who have brought forward current state-of-the-art approaches to genomic sequencing and information. Hopefully this will work. No. There. Okay, so what we'll do is to go through sort of a background for this information, the uses of markers, the significant risk, the IDE and FDA pre-sub process, which a lot of people do not understand, and the need to partner with assay developers. We've had a mixture of clinicians and assay developers speak. One of the problems with that is that oftentimes the two groups are distinct and different. As you all know in this room, in 2010, the IOM did have a first outline for reinvigorating the NCI Cooperative Clinical Group Program, and recommendation 7 was that the NCI, in cooperation with other agencies, should establish a consistent dynamic process to oversee the development of national unified standards as needed for oncology research. You're certainly hearing a number of aspects of that. We in the Cancer Diagnosis Program have tried to work with the FDA. We cannot speak for the FDA, and I do want to get that caveat out today, but that we have tried to bring many of their principles to the attention of investigators who are moving their discovery-based markers into clinical use in trials. And a lot of this has been driven, as you know, by documents that have come out in the lay and scientific press highlighting the risk for patients that are posed by new and poorly validated diagnostics. Trialists and investigators want to use markers. We've had 3 outstanding examples of this with positive results, but they oftentimes have not understood the rigors of clinical assay development, although the 3 previous speakers have all spoken to those issues. The FDA Office of In Vitro Devices also began in late 2010 to enforce its oversight authority over the safety of diagnostics used for medical decision-making in clinical trials. Need help with the slide. There we go. So we've had integral markers, and this is what we will really talk about. These are markers that are essential for performance of the trial. They are used for medical decision-making and specimen donor. The results are given back to the patient or physician, and these types of markers include eligibility criterion, the types of markers that have been mentioned by the previous speakers, treatment assignment, risk stratification, dose modification, and they must be performed in a CLIA-approved laboratory by law. The other markers are really research-type markers. The classes of markers are known to this audience, but prognostic, predictive, and pharmacogenomic markers are the primary ones. Certainly, the previous speakers have been addressing the use of predictive markers primarily as eligibility criterion, and the examples of such are clearly identified, known to you all: EML4 ALK fusion in non-small cell lung cancer. We've also had discussion of certainly the use of erlotinib in EGFR mutant and non-small cell, as well as these basket trials that might use erlotinib in similar EGFR mutation in other disease sites. But there are other markers that are also used for risk stratification, such as FLT3 internal tandem duplication, the PM1, CEPB alpha mutations in AML that can lead to, and certainly in the pediatric age group, to a stratification with a plan for early transplant, which may itself include a high degree of risk, such as 30% mortality. And also there is a case, again, in pediatric AML, where a busulfan pharmacokinetic assay is being used for the dosing of the specific patients on trial. This brings up the issue that has yet to really come up, which is the significant risk issue, which is largely essential to the FDA CDRH review of devices, or diagnostics are considered devices by the FDA. And the investigational device exemption regulations, which are cited there, require that significant risk device studies follow all of these regulations. And in general, a significant risk device is defined as a device that is, among other things, used for use of substantial importance. I would like you all to think about what is substantial importance. And this is one of the things that perhaps from this workshop we can consider. What does this particular term mean in the context of diagnosing, curing, mitigating, or treating disease, or otherwise preventing impairment of human health? It otherwise presents a serious risk to the health, safety, or welfare of a subject. Now, the regulations are really independent of whether the device is to be marketed commercially or only as an integral marker in a trial. However, significant risk is really not well defined or quantitated in several guidances from the FDA, including the information sheet guidance for IRBs that actually have to assess such risk. The draft guidance to IRBs and IDE addresses what and who to examine, but not how to examine risk. This is the one that was released recently in November of last year. And the draft guidance on companion diagnostics contains in footnote 5 that a, quote, device with a therapeutic product allows the therapeutic product's benefits to exceed its risk. That's probably the best definition of all for comparing benefit to risk. Perhaps this principle and how to measure it should be expanded in a separate guidance dedicated to risk. So what is an IDE? We have these wonderful examples of markers that have been identified, but an IDE actually allows an investigational device to be used in a clinical study in order to collect safety and effectiveness data. That is its purpose. The investigational use includes clinical evaluation of certain modifications of new intended use of legally marketed devices, and all clinical evaluations of investigational devices, unless exempt, must have an approved IDE before the study is initiated, and IRB may approve a non-significant risk investigational device exemption, but any IDE that has a question of a significant risk really does need to be reviewed briefly by the FDA. Now, PIs really need to explain the potential of a marker for risk in a protocol. What is the risk to the patient of a false positive or a false negative assay result? And this requires, by the PI, understanding the analytical performance of assays, the accuracy, reproducibility, precision, how these characteristics translate into false positives or negatives, and the analytical data will be different depending, as was shown in the previous talks, on the type of device or technology, DNA, gene expression, immunohistochemistry. This should be provided, hopefully, by a collaborator from the clinical lab. Is the risk of the device or marker less than the benefit of the treatment should be clearly articulated. A non-significant risk IDE can be handled just by the local IRB. But if it is, then it has to be documented in much the same way as the FDA wants to document IDEs. Finally, there is the formal pre-submission program, meetings with FDA staff. There is this draft guidance that is identified here. Hopefully these slides are shared with everybody, that outlines their current recommendations about clinical assay development and how to contact the FDA office. Now, once you've got to this point and you've considered that you have a significant risk device, then one goes into a pre-sub with the FDA. And this actually has been recently changed. This was a lessons learned document that we created with the help of the Children's Oncology Group, who were the first to go through this process for the 3 genetic markers as well as MRD by flow cytometry that were described earlier in this talk. But the pre-IDE review can take roughly 60 days. Recently, they've extended that time to 75. The IDE application itself, if one is necessary, is just like the IND and takes 30 days, and that is actually a legally mandated time limit. As you can see, the FDA looks for risk assessment, the elements of the trial document to be reviewed, an assay assessment, and then other things that are expected but not currently present in LOIs or concepts that also they want to see if you're going to submit this early. This is the Clinical Trials Network implementation, and one of the things that is important to realize is that this is an unfunded mandate in that the FDA clearance is independent of the Operational Efficiency Working Group timelines, which are outlined here. These timelines are being compressed somewhat from when this paper was written, but the importance of this is that as soon as an assay is going to be used in a trial or proposed, even at the concept stage, it is important to be submitting information about the validity and performance of that assay as soon as possible. Now, the Cancer Diagnosis Program does, in fact, try to assist this process. We have the Clinical Assay Development Program that is led by Dr. Conley, and we have also created templates that are freely available for immunohistochemistry, FISH, as well as somatic mutation documentation of assay performance. Now, there is one integral marker that is not a significant risk, and what we have come in several reviews now, and this is, pardon my Mac to PC translation here, but when a marker is used to balance randomization and it is not used directly for assignment of therapy, but rather to assure that patients have an equal chance for receiving a treatment, that is a non-significant risk situation. And to date, the FDA has allowed this to be carried forward as a non-significant risk. Now, one of the things that we've not really talked about in the 3 previous talks, and it'll be interesting in the discussion to get to, is a number of issues that we have seen in pre-IDEs now for CTEP trials that involve the accuracy, reproducibility, interfering substance analysis, stability of the analytes, the linearity of an assay, limits of detection, recurrent reference interval, and measurement bias. These are, the FDA depends upon the Clinical Laboratory Standards Institute documents that are referenced here, but one of the problems, I think, for a lot of the tests that we have heard about that are extremely exciting is how reproducible are they? Is the information really as stable as one might want? And the orthogonal assays that were alluded to actually by Dr. Miller may be extremely important to assure that the result is accurate. So the recommendations for protocols for PIs with markers would be wise to include a section in a protocol. What is the risk of the false positive or negative results? How are the benefits of treatment greater than the risks? Is in their mind an IND or IDE required? It is extremely important that we remember that in the patients that were preceded and the use cases that were given in the 3 preceding outstanding talks, the context of disease is extremely critical, and this does come into the FDA consideration. In all of those cases, these were patients with advanced disease who really had no other options. And so when you think about balancing risks, that's got to be taken into account. It is a process that is continually a work in progress. In the informed consent, one of the things that we have clearly seen from these reviews is that the FDA does want to know exactly what patients are being told about integral markers and the consequences of assay results. It is also helpful to make sure that the assay developers are partners and collaborators in the trials, and the 3 previous speakers all clearly are doing that, as well as members of the IRB. So in summary, protocol investigators, assay developers, performers, and sponsors really need to collaborate and partner closely. The 3 previous talks really spoke to that in spades. If trial has an integral marker, then an IDE may be needed, and IRB investigator and sponsor need a pre-IDE submission to the FDA. Can be hard for the FDA and the IRB to find the risk attendant to an integral marker and need to know how to define that those rates and what that means for toxicity to a patient. Both the IRB and FDA need to know what patient is told about the risk for patient in a consent. And one of the things that it would also be very good if this workshop can address is whether this increased attention to the development of molecular diagnostics improve the quality of care. Thank you very much.